Hello, I'm Sanjana.
And I'm Jesus.
And I'm Jillyn.
We are from Period 5 AP Biology, and this is our FastPlant e-journal.
FastPlants were developed by Professor Emeritus Paul H. Williams for the purpose of disease resistance in breeding.
These Brassica rapa plants were selectively bred for their speed in growing, ability to produce seeds, small size, and facility of growing.
Professor Williams bred and grew numerous generations of the plants, selecting adults to pollinate based upon his desired characteristics. After 20 years of this process, the 6-month life cycle was reduced to a 5-week cycle.
The life cycle of a fast plant is similar to that of a normal plant, except it takes a fraction of the time. During the first 2 days after planting, the seed germinates and pokes through the soil. Roots and shoots emerge.
During the next 9 days, the plants grow and mature. Leaves, hairs, and buds develop.
During the next 3 days, the fast plant flower and flower structures and functions develop.
During the 3 days after that, pollination and reproduction take place. Because of the lack of bees and other natural aids of reproduction, students have to pollinate the flowers themselves.
During the final 17 or so days of the life cycle, a new genetically unique single cell is formed. The cell turns into a seed through embryogenesis. The original fast plant dies.
And now for a bit of background on evolution and artificial selection. Evolution is also commonly known as descent with modification, a phrase Darwin used in proposing that Earth's many species are descendants of ancestral species that were different from the present-day species. In more specific terms, it is the genetic changes of a population from generation to generation.
Evolution was once described by Lamarck's theory of acquired traits. Basically, this theory stated that any quality acquired by an individual during their lifetime can be passed down through its offspring. Clearly, this was inaccurate, since evolution is genetic-based.
Our modern knowledge of evolution is fundamentally based on Charles Darwin's findings. Darwin's famous studies of the finches in the Galapagos Islands led him to notice that the characteristics of similar species varied somewhat from place to place. This is where he got his term, descent with modification.
He explained this phenomenon with natural selection. Natural selection occurs over generations in which favorable traits, which enable organisms to have a survival advantage over others, tend to dominate in the decades to come. This leads to evolutionary change.
Artificial selection abides by the same principles as natural selection. However, instead of nature dictating what the favorable traits are, humans choose them.
Artificial selection originally started off with modification in crops and livestock. For example, the modern corn comes from an ancestor that looks similar to the teosins. Over generations, it has been artificially selected for the oil content of its kernels.
With the development of artificial selection, different breeds of domestic animals have been created. For example, greyhound dogs resulted from selection for maximal running ability, which is the reason why greyhounds have long legs, a long tail for balance, and arched back to increase stride length and great muscle mass.
Finally, here is some information on trichomes. Trichomes, from Greek meaning hair, are cellular outgrowths of the stems, leaves, and reproductive organs of plants, algae, lichens, and certain protists. They have multiple functions such as deterring herbivore attacks and radiation, and preventing infection, extreme temperature, and excessive evaporation. Essential oils from the trichomes can be found in herbs used for cooking.
Our experiment was performed to answer the question, can fast plants be artificially selected for hair? In other words, can human nurture affect the growth of trichomes in these plants? Or is the amount of hair dependent simply on nature, or the genes of the plant?
Our hypothesis was that fast plants can be selectively bred to produce more trichomes.
To set up this experiment, first fill a pot completely with soil. Water the soil until it is completely wet in order to make it more compact, and let it drain through the holes of the bottom of the pot.
Place 8 seeds on the surface of the soil in a flower shape with one in the middle, and use a pencil to gently push each seed approximately 1/8 of an inch below the soil. Spread the soil evenly on top and place the plant in a fancy school greenhouse where artificial sunlight can shine on it 24/7.
Water the plant whenever the surface of the soil seems dry, and fertilize it every week or so to facilitate growth. Measure the plant's height every other day once the seed germinates.
Once flowers have developed on the fast plant, use a Q-tip to pollinate random flowers. Simply touch the Q-tip to a bud to collect some pollen, and then touch the bud of another flower to transfer the pollen. Throw away the Q-tip if you value your life, or at least your AP Bio grade.
About 3 weeks after planting the fast plant, use a magnifying glass to count the trichomes or little hairs on the plant. Record data.
Once all trichomes are counted, locate the seed pods on the plant, detach them, and roll them between your fingers to take out the seeds. Save the seeds for generation 2 according to losers, few trichomes, and winners, many trichomes. Dump out the plants and soil and clean the pot.
Here are some pictures of our plants throughout their life cycles.
These are our 22nd day statistics.
This data table and the accompanying histogram show the number of plants with each range of trichomes in the first generation plants in both periods.
Some mistakes that were made in the first generation were that we didn't prepare for the heat wave, our soil may have been inadequate for growing fast plants, and we didn't pollinate enough, so the plants didn't create enough seeds.
In the second generation, we plan to water more during heat, pollinate more daily, and not push seeds as far down into the soil when planting.
Thanks for watching and have a great day!